Welcome to the Two-Way ANOVA tutorial. Today we're going to be talking about the Two-Way ANOVA, how to run it in SPSS, and specifically how to run it for the EMG lab. So a little bit of a review. When we're thinking about a one-way ANOVA, which is the more basic form of the ANOVAs, we have to consider that the goal of the ANOVA is to distinguish or compare means and determine when you have two or more, whether there is a statistical significance between the three or four or five. And thinking about an experiment, let's say we were simply doing an EMG experiment in which we determined or measured the EMG responses under different weights. So this would be a perfect scenario in which we're running an ANOVA because it would actually allows us to say, hey, is any of these weights different than the rest? Now, we have a slight complication, and that complication is the fact that we don't only have different weights, but we actually had handedness. So, in the case of this particular experiment, we have the low, medium, and high weight, but we also have the dominant and then the non dominant hand. And this creates a complication because we not only can test the weight, because there is a second factor embedded in it, but also we can't simply test the hand because we have this embedded factor of weight. So this is where the two-way ANOVA comes in, where you can measure two factors, in this case, the weight and the hand, and determine whether the hand or the weight or the combination of the hand and the weight has a particular significance in this model. So once you finish your lab, you should have filled up all these little tables. The trial number, it's already in there, and the peaks would be the peaks that you got from your analysis from MATLAB. So these all should be filled in for both the hand, the dominant hand, and then the non-dominant hand. Now, sorrowfully, the stat software requires you to format it in a very particular way. So the way that you're going to do it, and my recommendation is to do this in Excel, is to basically do the trial, hand, weight, and then the peak value. And this is this data from the sample EMG data. And for the entirety of the dominant hand, you're going to do the 10 samples for black, the 10 samples for gray, and then the 10 samples for orange. Once you do those 30, then you're going to go into the non-dominant hand and do the 10 of black, the 10 of gray, and then the 10 of orange. So it shouldn't be that hard copy pasting around from Word to Excel. So you can do that. Once you have that, what we're actually going to do is copy paste this into SPSS. So once you select everything, copy, you can go ahead and click here, paste with variables. And what that will allow you is to paste not only the words and everything, but also the headers. SPSS is very quirky when it comes to copy and pasting, so just be careful about that. Now, to run a two-way ANOVA, we need to go into Analyze, General Linear Models, and then Univariate. And this is going to ask us what the dependent variable is, which in this case is the response. What value are we measuring? And then the fixed factors, in this case, there's only two. It's the hand and the weightedness, because I'm running a two-way ANOVA. So I'm going to go ahead and add them here. So I have my peaks for dependent variables and my hand and weight as fixed factors. In the case of plots, we're going to actually go ahead and generate some plots. We're going to set the hand as horizontal axis and the weight as separate lines and add it. And then we're going to do the reverse just to see what happens when you actually differentiate between the. So having now the hand and weight and hand for the plots, we're going to go ahead and continue. And then for post hocs, what I'm going to go ahead and do is do weight and select a post hoc test for it. And we're going to go ahead and click this two keys. It might not be clicked when you run it, so make sure that you click it. What two keys is going to do is that, remember, ANOVA's hypothesis is to simply determine whether there is at least one difference between the factors. So it's going to tell you, well, at least the black between the black, the gray, and the orange, at least one is different, but there could be two that are different. There could be all three that are different. So it doesn't differentiate between the differenceness, if that's even a word, between these factors. So what Tukey's does is that it actually compares each of the factors to each other and determines whether there is a significant difference. And we'll see the results to make a little bit more sense. So having the plots and the post hoc, we're just going to go ahead and run. The first output that we're going to see is this ANOVA table, or this test between subjects effect. And what this is telling me is that, particularly for this little column here on the significance, is that the hand is significant. It is less than 0.001 of the p-value. The weight is significant, meaning that the weight also has an effect. But interestingly enough, and this is one of the benefits of the two-way ANOVA, is that it tells me that the hand and weight combined together also is significant. So what this means is that the handedness of the subject is important, the weight is important, it's differentiable, but also the combination between the hand and the weight also matters in interpreting the results. So this means that those two combined effects could be also considered a third effect, which is kind of like an additive effect that is nonlinear, meaning it doesn't simply add. Now for the post hoc test, specifically to the weights, and you might ask, wait, why didn't you run it for the handedness? And the reason is that SPSS doesn't like to run Tukey's for only two factors. So for the post hoc test, we'll see that all of them are significantly different. So black to gray, black to orange, gray to black, gray to orange, orange to black, orange to gray. This table is a little bit redundant, but I can see that throughout all the comparisons, there is a statistical significance. So what that means is that all the bands of different colors are being detected differently in the EMG peaks. Personally, I prefer this table of summary rather than this one. And what this is basically telling you is the subset is any variable that it is within one column is part of a group, which means that it's statistically significantly different from any other variable in any other group. So if I look at this, I am basically confirming what I see here, which it means that orange, gray, and black are their own groups, meaning that they're all statistically significantly different from each other. So orange is here, gray is here, and black is here. And you can see that the average mean or observed mean is lower for orange, a little bit higher for gray, and then greater for black. You can ignore the significance. We really don't pay attention to this significance. It's just the fact that the groups are made. So that's the second part of what you're going to be doing for your post lab. The third part is simply getting these tables. Now, these are estimated marginal means, which are a little bit complicated. I'm not going to go too much into detail for what this exactly means, but it is a way to balance all possible comparison points. So it takes into account the handedness, the interaction, the weight, and some other covariates that might be present in the model. So it might not necessarily make logical sense because right now you see that the dominant is lower than the non-dominant. And if you think about it, that makes no sense. But what I am looking for in these plots is the effect size. So what that means is the slope of this particular line. And I can see that, for example, for the orange, I see that there is a very strong effect size, meaning difference between those two factors compared to the gray. And the black is kind of in the middle. So that is very interesting when you have a low weight or a high weight. The dominant and the dominant become a little bit more easily to differentiate than when you have the gray. And I can do the same for the bands, depending on the dominant and non-dominant. So I can see that, for example, the slope between black and gray for handedness, for the non-dominant hand is very large. They're all pretty large going down. So this is a good way to measure the effect size or the impact of each of the variables in your model. So you're going to be doing plot, plot, Either of these tables is fine for the post lab, whichever you understand best. They're telling you the same information. And then this ANOVA table here. So if you have questions, let me know, but that should conclude the two-way ANOVA tutorial.